Enzymes will not be always in an activated state. So only when their function is needed or when their job is needed, then only the enzymes are activated. Proteins, when they're subjected to higher temperature, they get denatured. Transferases means what? Catalyze transfer of a group between a pair of substrate. Coenzymes, organic compounds which loosely bind with enzyme. Hello everyone, welcome back to session 6 of this chapter called Biomolecules. So in the last session we were studying about enzymes. So what are enzymes? Enzymes, as we know, they are biological catalysts which speeds up a chemical reaction. And we all know that the function of enzymes are so very important for a biochemical reaction. And we also know that the enzymes are specific in their action and they exhibit a very important property called enzyme specificity. Enzyme specificity. Enzyme specificity. What do you mean by enzyme specificity? Enzymes are specific on acting on a particular substrate. For example, like carbohydrates are acted upon by carboxylases. Proteins are acted upon by proteases. Lipids are acted upon by lipases. So that is called enzyme specificity. And enzymes work in a lock and key system. Like, what do you mean by lock and key system? For a particular lock, to open a particular lock, we use a particular key. So in a similar way here, the lock is nothing but the substrate and the key is the enzyme. So to act upon a particular substrate, a particular enzyme is used. So that is called enzyme specificity. So what is the nature of enzyme action? Steps involved in catalytic cycle of enzyme action. I think all of us know enzymes will not be always in an activated state. So only when their function is needed or when their job is needed, then only the enzymes are activated, and there are certain activators in the body which is going to activate that particular enzymes. For example, like all of us know HCl, hydrochloric acid, that is produced by the gastric glands. You know, this HCl will activate inactive pepsinogen, and inactive form of enzymes are called zymogens. The inactive forms of enzymes are called zymogens. So this zymogen, which is an inactive enzyme, is activated into active enzyme. It is activated into active enzyme. Now let us understand, as I said, the enzyme action works in a lock and key system. Enzyme action works in a lock and key system. Here, substrate. Here the substrate might be a carbohydrate, it might be a protein, or it might be a lipid. So substrate is called as a lock and enzyme is called as a key. So this is going to act upon a particular substrate. That is what we call it as enzyme specificity. As I already said, the carbohydrates are acted upon by carboxylases. Proteases don't act on carbohydrates. Then lipases act only on lipids. They don't act on proteins nor on carbohydrates. That is called enzyme specificity. Enzymes are specific in their action, and that is called as enzyme action, which can be compared to a lock and key system. So here you can see the beautiful picture, the principle that is involved in the enzyme action. So here you can see the enzyme and the substrate. So here you can see the enzyme plus substrate. So when an enzyme combines with a specific substrate, it forms enzyme-substrate complex. And I think all of us know enzymes are neither used up in a chemical reaction nor their structure is altered. You get back the enzymes as it is and it can be reused again and again. So that is another very important feature of enzymes. Neither they are used up in a chemical reaction nor their structure is altered. So here, and it forms enzyme substrate complex, and this enzyme substrate complex splits again into enzyme and product. So this enzyme will get back into what? this enzyme will be recycled to carry on the further functions, and we get back the product. So this is called the enzyme specificity. So when a specific enzyme acts on a particular substrate, it forms enzyme substrate complex, and this, when it splits, it gives back the enzyme and the products. So the substrate binds to the active site of the enzyme, fitting into the active site. So for example, like to open a particular lock, do we use any other key? No, there is one specific site where it will help the key to open. So in a similar way, there is a specific site on the substrate where the enzyme will fix. So that is what the substrate binds to the active site of the enzyme, fitting into the active site. Like how we cannot open. So if it was so, I think there would not have been a concept of lock and key system, right? For any lock, we cannot use any key. It will not open. So there is one specific arrangement that is made so that that particular key will fit into that particular lock. So in a similar way here, the substrate will have particular site, and a particular enzyme which acts on that particular substrate, it will fit into that substrate. Then the binding of the substrate induces the enzyme to alter its shape, fitting more tightly more around the substrate. See how it works. So there is one specific site. For example, if this is a substrate, see, this is how. So this is a substrate, and this is the enzyme. See how it fixes. So perfectly it is going to fit into the particular site of the substrate so that it can act upon. And then this is called as what? Enzyme substrate complex. This is called enzyme substrate complex. So active site now breaks the chemical bond of the substrate, and enzyme product complex is formed. So the enzyme releases the product. So this is all about the enzyme action. So as I said, enzymes are nothing but biological catalysts which speeds up the chemical reaction. For example, as I said, morning we have a breakfast, by around after 2 hours we feel hungry. So what has happened to the food? All the food is digested in presence of enzymes. So enzymes are very, very important. You can call them as biomolecules for the process of digestion and for various physiological process, may it be in plants or in animals. So the enzymes work in a particular lock and key system where for a particular substrate, a particular enzyme acts upon. So here, this is how the picture is shown, the enzyme action. Here enzyme is acted upon by, for example, if the carboxylases, these substrate is carbohydrates. They are acted. So it forms what? Enzyme substrate complex, which releases back what? Enzyme and the product. So this enzyme can be used again and again. As I said, enzymes are neither used up in a chemical reaction nor the structure is altered. It can be used again and again. So the substrate binds to the active site of the enzyme, fitting into the active site. Then the binding of the substrate induces the enzyme to alter its shape, fitting more tightly more around the substrate. Then active site now breaks the chemical bond of the substrate, and enzyme product complex is formed. The enzyme releases the product. Then what are the factors that affect enzyme activity? I think all of us know most of the enzymes are proteins, but all proteins are not enzymes. And very important is what? Temperature. I think all of us know proteins, when they are subjected to higher temperature, they get denatured. So as we know, the enzymes are proteinaceous in nature. The temperature definitely has a very important it is a very important factor for enzyme activity. Now let us understand what are the factors that affect the enzyme activity. First is temperature, next is pH. So show highest activity at optimum temperature. Activity declines above and below the optimum value. So the optimum temperature is very important. So it can work, enzymes will work in a particular optimum temperature, and above the optimum temperature, it will get denatured, and below the optimum temperature, it will not work. So temperature has a great role to play in enzyme activity. So temperature show highest activity at optimum temperature. Activity declines above and below the optimum temperature. Next, pH. Enzymes always function in a narrow range of pH. Highest activity at optimum pH. So the optimum pH. So in optimum pH, the activity will be more. So below that narrow range of pH, no, it will not work. So temperature and pH have a great role to play in the enzyme activity. So what are the factors affecting enzyme activity? One is temperature. As we know, show highest activity at optimum temperature. Activity declines above and below the optimum value. pH, enzymes function in a narrow range of pH. Then highest activity at optimum pH. Concentration of substrate also is one very important factor. So what do you mean by So as we know, the velocity of enzymatic reaction rises with increase in substrate concentration till it reaches the maximum velocity called Vmax. So further increase of substrate does not increase the rate of reaction as no free enzyme molecules are available to find with additional substrate. So very, very important. Another very important factor that affects the enzyme activity is the concentration of the substrate. As we know, a particular enzyme acts on a particular substrate. So the velocity of enzymatic reaction rises with increase in substrate concentration. So till it reaches maximum velocity. Further increase of substrate does not increase the rate of reaction as no free enzyme molecules are available to find with additional substrate. Now, enzyme inhibition. As I said, do you think always round the clock the enzymes are activated? No. What will happen if round the clock the enzymes are in an activated state? It is going to engulf the cell itself. So, only when their function is needed, then they are activated. So, there are enzyme. So, let us understand about what is enzyme inhibition. There are some inhibitors which is going to stop the enzyme activity when its function is not needed. Just like how enzyme activators are there, there are also enzyme inhibitors. So, now let us understand what is enzyme inhibition. When the binding of a chemical shuts off enzyme activity, the process is called inhibition and chemical is called inhibitor, and the chemical is called the inhibitor. So once the enzyme activity is completed, so it has to stop functioning. So a chemical will come and bind to that particular enzyme and it will stop the enzyme activity. Such chemicals which stop or bind to the enzyme after their enzyme activity are called as enzyme inhibitors. For example, here you can see we have this two types: competitive inhibitors, non-competitive inhibitors, uncompetitive inhibitors. So when the binding when the binding of a chemical shuts off enzyme activity, a chemical, when the enzyme activity is totally completed, so it has to stop. The enzyme activity has to stop. So what is that which is going to stop the enzyme? What will happen if the enzyme activity is not stopped and if it continues? So it is going to damage the cell itself. So in order to protect the cell, so there are certain inhibitors which is going to which are nothing but chemicals which is going to come and bind the enzyme and stop the process of the enzyme activity, and such and that process is called as enzyme inhibition. So what is competitive inhibition? As I said, we have this competitive inhibitors. So inhibitor closely resembles the substrate in its molecular structure and inhibits the enzyme activity and competes with the substrate. So inhibitors closely resembling the substrate in its molecular structure and inhibits the enzyme activity and competes with the substrate. For example, inhibition of say a succinic dehydrogenase by malonate. So this is how the enzyme activity is suppressed or stopped. Such inhibition is called as competitive inhibition. So what is competitive inhibitors? Inhibitor closely resembles the substrate in its molecular structure and inhibits the enzyme activity and competes with the substrate. Classification of enzymes. So now let us understand what is the basis for class. Based on the type of substrate they are going to act upon, they have been broadly classified into different types of enzymes. For example, we have the first type called oxidoreductases, second one is called transferases, third one is called hydrolases, fourth one is called lyases, and fifth one is called isomerases. Then ligases. All these are the different types of enzymes. As I said, what is the basis for classification of enzymes? Based on the type of substrate they are going to act upon, they have been broadly classified into oxidoreductases, transferases, hydrolases, lyases, isomerases, then ligases. Now let us understand one by one. So here you can see the beautiful picture. So oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases and translocases. Now, moving on to oxidoreductase, the very word itself will tell you. They are also called as dehydrogenases. Catalyze oxidoreduction between two substrates, hence the name oxidoreductase. So, oxidoreductase or dehydrogenases are the enzymes which is going to catalyze or catalyze the enzymatic activity called oxidoreduction between two substrates, hence the name oxidoreductases. Transferases, the very word itself will tell you. Transferases means what? Catalyze transfer of a group between a pair of substrate. Then hydrolases, the very word hydrolyze means what? Breaking. So catalyzes hydrolysis of ester, then ether, peptide, glycoside, then PN bonds. Then lyases catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving a double bond in the product. Isomerases catalyze interconversion of optical, geometric or positional isomers. Ligases, the very word itself, as we already come across, ligases act as molecular stitches. They are going to join. So, catalyze linking together of two compounds. Right. So, what is the classification of enzymes? Enzymes have been broadly classified based on their action. Then oxidoreductases are based on the substrate they are going to act upon. So, oxidoreductase dehydrogenase catalyze oxidoreduction between two substrates. Transferases catalyze transfer of a group between a pair of substrates. Then 3 hydrolases catalyze hydrolysis of ether, then ester, ether, peptide, glycoside, etc. Lyases catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving a double bond in the product. Then isomerases catalyze interconversion of optical, geometric or positional isomers. Ligases catalyze linking together of two compounds. Then moving on to the cofactors. Here you can see apoenzyme and cofactor together form holoenzyme. So protein portion, and this is protein portion is called apoenzyme. So it is the protein part. Protein part of an enzyme is called apoenzyme and the non-protein part is called cofactor. The non-protein part of an enzyme, that is non-protein part, is called cofactor. So apoenzyme plus cofactor form a holoenzyme. Here you can see the cofactor. So what are cofactors? Non-protein constituents found to the enzyme to make it catalytically active. Protein portion of the enzyme is called apoenzyme and prosthetic groups are organic compounds tightly bound to apoenzyme like hemine, peroxidases and catalase. Then coenzymes, organic compounds which loosely bind with enzyme. Example NAD, then nicotinamide, adenine, diphosphate. Then metallions required for enzyme activity from coordination bond with side chains at active site and with substrate. Example zinc. Zinc is a cofactor for enzyme which enters the stomach. So non-protein constituents and the protein constituents. So here protein part of an enzyme is called apoenzyme and cofactor is a non-protein part which forms a whole enzyme active. Hope you have understood all the concepts what I have explained in this particular chapter called Biomolecules. So I'll be in the coming session with some more interesting concepts of the chapter, the new chapter in the coming session. So I'll be back with some more interesting concepts in the coming session. Till then, goodbye and thank you.